a liver macrophage.
So in the sinusoid.
Which we call a Kupffer cell. That's the name it primarily goes by instead of liver macrophage. But remember, most macrophages are in loose connective tissue. We talked about the alveolar macrophage being in our alveolus, just under the surfactant. And here we have a macrophage, usually it's just monocytes that we find in the blood. But the purpose of these macrophages is to help to break down the old red blood cells. And then the liver processes their pigment, the bilirubin. And it takes that bilirubin and it adds to it bile salts and secretes that through into the bile ducts. So that's our third structure of our portal triad, is a bile duct. So we'll add that in here. Bile ducts have simple cuboidal epithelia. So it makes it a little bit easier to distinguish them from the wide diameter portal vein and the muscular smooth muscle artery. And so, I know I can do this easier than you can. I'll just do it part way here. And so coming between the hepatocytes, we have the small channels that carry the bile, and these are known as bile canaliculi. So remember we have the canaliculi in bone tissue? So it's the same word, but a different structure. So this is a bile canaliculus. It is not a separate tube. Like a capillary has a simple squamous epithelium wrapped around it.
Just two notches.
It's really two spaces. So you guys stand up, Stacy, and go back to back. Now, height-wise, maybe you're not quite right. Now, notice how we have the secondary curvature, the lumbar curvature, and I can put my hand through there and actually not touch their backs too much. So Stacy's one cell, Chuck is another, or Charles, or whatever. So you can sit down now. So that space where there are two not basement membranes, but basal membranes were not joined by tight junctions, creates a small channel. You add two more cells up to that, and two more cells, and two more cells, and we get an elongated tube. And that's what the bile canaliculus is. All right, so if I were to diagram that on the board, which I will do, so I'll leave that there. Now, you erase this sequence here. You guys have that?
Not yet. Thank you.
Okay.
Okay, so this is called the
So here would be a sinusoid. And here would be a hepatocyte. They don't have microvilli, but they have what are called microrugae or microfolds. That's not a term you need to know. And then here would be this gap and another cell here, tight junctions, but that would be the bile canaliculus. Okay?
What's the purple thing?
That's the sinusoid.
That's the basal membrane?
It's not a basement membrane, it's just the bottom membrane of the cell. So this would be the apical surface, this would be the basal surface. Okay, top and bottom of the cell. Same thing here. And so our bile secretion would be here. All right. Now there's a bit of a space here between the epithelium of the sinusoid. This is just simple squamous epithelium. This is like a large capillary. It's a wider diameter than our typical capillary. And the space between the two is known as the space of Dissay. So glucose is going to be transported out of the sinusoid into the space, and then the cell's going to take it up and either utilize it or store it as glycogen. Same thing with amino acids. You guys know how I like electron microscopy, so we have, I'll come back to this bottle in just a moment. So here's light microscopy view of the central vein, and you can visualize parts of the sinusoids traveling towards the central vein. The next slide is of a really nice slide of a portal triad, one of those 6 corners. So here would be the portal vein, thin wall, large diameter. Here would be the hepatic artery, smaller diameter but more smooth muscle. And then this would be part of the bile duct with all the nuclei, simple cuboidal epithelium. The rest of these are probably lymphatics or branches of those vessels. But those 3 structures make the portal triad. There's a lot more connective tissue around them than we find around the central vein. And then again, here would be the sinusoids traveling by the hepatocytes. So here's a view of a hepatocyte. Down here would be the sinusoid with its simple squamous epithelium. All right. Here's a bunch of mitochondria right there at the surface, and this is the space of Desay. So these are not microvilli, but tiny folds, or rugae. Here's the opposite end of the hepatocyte. More mitochondria. A lot of ribosomes and glycogen, stores glycogen. Here's the tight junctions, and here's the cross-section through a biocanalicculus. At the opposite end. Now, what 2 major sets of organelles would you also expect to see in a liver cell given its function, Golgi and ribosomal ER, because it's doing a lot of protein synthesis. But remember, you don't want to drink and take a lot of Tylenol at the same time, because the liver functions to break down toxins. It breaks down Tylenol, it breaks down alcohol, and it's really bad for the liver to do both of those together. And what organelles do we use in detoxification? Smooth ER. So the liver is unique. It has both a lot of ribosomal ER and a lot of smooth ER because of those 2 major functions. Okay, again, we'll come back on Thursday when we talk about digestive physiology and talk about the hormones and enzymes. So if there's, the liver is important in releasing the bile salts in the bile. So if there's a lot of fat in the diet, hormones are released and nerves like the vagus nerve are activated to cause the liver and the gallbladder to release more bile. Okay, so that type of control we'll talk about on Thursday. But I want to come back and finish up with the pancreas and the bile ducts leading to the pancreas for about 5-8 more minutes. And then we'll be done with lecture for the day. So can I erase the board? May I?